So as Simeon essentially already said, the accelerator trials are an important legacy of the GCFF program, and they are going to be a place for ongoing research to make the best use of these sites. Whenever possible, we're aiming to use these interventions not just to improve the current crop, but again, as Simeon said, to improve the site itself so that we're going to be benefiting future rotations as well as current rotation. We're also committed to maintaining strong industry involvement in these trials and working with forest managers to develop future interventions over time. So all in all, these trials are a big, audacious challenge for Scion and the sector, but they are already providing value, and they will continue to provide value for the next 25 years. All right, first question. How do I get hold of Bio-Ret for every tree I manage?
We're working on that. Yeah, okay. There's a limited amount I can say about that, but we are very actively working to get it made in New Zealand. The stuff we've worked with on operational scales, we've imported from China. The cost of production over there was minimal as it would be here realistically. It was just simply getting it from there to here. But there's a lot of issues to work through, but we are actively working down that pathway.
Okay, let's do the related question. Are you testing soil toxicity for using Bio-Ret on short, mid, and long term?
We've got as part of that work to make it commercially available in New Zealand, there are a bunch of EPA regulations to move through, and again, we're working through those processes. Again, it's just part of the package of making it available here.
Okay, where to with all this work, given GCFF has finished and the latest MBIE funding bid was not successful? Who wants to take that one?
Well, as someone who's gone over to the industry, I think as we've seen these long-term trials, very fundamentally important. And I think during my time at Scion, I sort of inherited a legacy of trials where I guess we hadn't probably invested a lot in them for a number of years. So I think it's important that there is continuity of funding. So, I guess hence the question. Obviously, the industry partners that host these trials, there's a lot of in-kind support that we get from them. And I think, Scion colleagues are working very hard to ensure that we don't lose these trials. At the moment, we have an interim program of funding supported directly by the Forest Growers Levy Trust for this exact purpose, to maintain that investment that's been expended so far, so that we can make sure that we have these trials as a legacy going forward.
Okay, one for Simeon and Sarah. Can Accc Deaminase be added to site soil tree through a spray application?
So generally we've sort of steered clear of trying to apply a microbe in this context. For bacteria, all of the functions that we want to see generally are out there in the forest. They're already there. It's simply about trying to encourage the behaviour you want to see, so it's 5% more often, 10% more often. And whether we've known it or not, we've been doing that anyway with our selection of genotypes, our breeding programs. It's no accident that trees that are generally tended to be more resistant naturally are really good at recruiting these bacteria anyway. All this activity is out there. We just have the ability to recognise that now, and we're starting to get tools to actively do something about it. I mean, it could be that there is some super bacteria we could add, but again, I say the capability is already there, and we can see it's responsive. It is sensitive. We can use that sensitivity for us.
Okay, one for John. Wouldn't thinning at a higher stand density increase the risk of wind throw?
So if you thin stands that are overstocked, then absolutely yes. And so that's why we want to use these concept of density management diagrams to help guide those decisions, so that we actually apply the thinning at the ideal time. So in the past, we've typically thinned on height, but when we apply these treatments to enhance productivity, we probably expect to see a diameter response. So we need to actually think about the degree of site occupancy at the time of thinning and make our decisions accordingly. So we can ideally optimise our thinning so the trees aren't too tall and skinny and therefore more vulnerable to wind throw. So yeah, if thinning is done poorly, then we could see increased risk of wind throw. But if it's done in a timely manner, we can have the opportunity to increase yield and manage that risk of wind throw accordingly.
Okay, we're going to keep you talking. John, what is the size of the prize? How much can we optimize individual sites across New Zealand? How much is this worth to New Zealand?
So I guess, sort of this concept of precision forestry is a balance between sort of site specificity and getting gain versus the sort of management complexity and the cost that goes with it. And I think there's sort of opportunity to try and identify the kind of management scale that is appropriate. So if we can sort of understand sort of broad sort of regions, sort of create management zones of appropriate scale, then we can really target our management. But I think, I'm in danger of being videoed and quoted here, but quite easily you could be looking at 20-30% gain in productivity. If you had a look at that earlier work, for a given level of 300 index, depending on the degree of site occupancy, you had pretty much a twofold difference in actual MAI just from appropriate or actual use of the site through stocking it with trees. So I think if we can look and raise, if that site carrying capacity, and instead of being 1,200, if it is actually 1,500 and we thin accordingly, then I can't see a reason why we couldn't be looking at sort of 20 to 30% gain. But that's what we're going to learn going forward.
Right. We'll give John a bit of a break. Loretta, for farm sites what will the second rotation productivity be if the first rotation was 18% and it was left untreated for the second?
Wow. Hello. Yep. We've got one example at Accc, if anyone's been there. It's just south of Rotorua. That is a pasture site that was planted in the 70s, and it did productivity that was astounding at the end of the first rotation. And it's now coming to the end of the second rotation, and it's again wowing everyone with its productivity. And that's because we kept what we've got on site. We basically harvested by keeping the residues on site. We kept the forest floor on site, so we looked after what we had. So if you look after what you've got on your site, you could do just as good. And if you improve your stock, so for example, if you're in an area that's got Dothostroma, if you plant a stock that is Dothostroma resistant, you will do better because your engine, which is your leaf, will be able to grow that tree. So, yeah, the answer is you can do better.
All right, back to John. In our ripping experiments, we generally found that growth after planting was more uniform with ripping in southern Kaingaroa than without ripping. Late in the rotation, mortality increased in unripped plots compared to ripped ones. This decreased growth late in the rotation. Enhanced canopy dynamics?
Yeah, I think it's an interesting one, that result that we saw in Rangipo, and I think there was a couple of reasons. One was the sites where the ripping occurred. We saw from the electrical conductivity mapping that there was some differences in soil moisture, and I think, as I mentioned in the talk, there was sort of a weed infestation. So I think the effects there on sort of canopy dynamics, where you get weeds, you will certainly have some suppression of trees. So we saw early differentiation. I think it's going to be interesting to follow those through. Some of the work I'm looking at now is to go back to some of those experiments where ripping has been done to look at what happens through to later rotation. So I don't have an immediate answer to the question, but it's something that I definitely want to look at going forward.
Right. So we still have lots of questions up here, but again, I want to give opportunity if anybody in the audience is not using the technology but has a burning question. Okay, we have one over here. Can we get a mic so that we can hear his question, though? Or maybe I'll just borrow a mic from somebody up here.
Are there plants or microorganisms that produce biuret naturally? Same for ACC Deaminase? I mean, do we have to export an industrial product? Can we grow it? Can we... yes.
For biuret, I would be, possibly, but I would be incredibly surprised if a plant is making something like that. It's a very high energy process. It's basically the same amount of energy that you need to make urea. It's not going to be forming naturally. on the Accc Deaminase one, a wide range of things can actually make it. I mean, we're working with bacteria, but fungal species can make it as well. There is some possibility that some plants can make it to help regulate their plant stress response, but from the responsiveness we see, I'm pretty confident that Pinus radiata is not one of those. But again, I don't know, but it's a possibility, whereas we're working with bacterial systems where we absolutely can see it, measure it, and see the response.
Okay, do we have any other ones from the floor? Ah, yes.
Following on from your response, I mean, it sounds like you're creating some sort of input to the forest. Oh, sorry. Just following on from your response, have you gone into the economics of this biuret and also the environmental ramifications?
Yeah, looking at that as absolutely part and parcel of the work we're doing towards determining commercial viability. Yeah, so some of you may even receive a survey in the near future touching on some of the issues of how much you would value certain things that we think biuret can do.
All right, going back to the questions up here. From the accelerator trials, are we at a point of identifying productivity gains between GF19, I think that's, and control pollinated stock in terms of mean annual increment growth?
So I think probably the accelerator trials, as we said, they're very young trials, but there are other trial series around, and I think two of them are the previous silviculture breeds trials, well actually three. There was the 1975 through 1982 genetic gain trials, but more recently the Radiata Pine Breeding Company have installed trials, large plot trials, to do just this, where they've put in stock with different levels of genetic improvement compared against a base seed lot of GF19, so that you've got comparison across all the trials. What we looked at and actually presented last year in the Tauranga conference was that if you compared what was the highly improved material, I think with a GF plus rating of about 25 to stock with GF plus rating of around 9, you were looking at about 25 to 30% gain in volume at the end of rotation and about sort of just over 30% gain, I think, in stumpage from memory. So the historic trials have given us that information, and the next generation of large plot genetic gain trials going forward will help us to understand the next generation of improved breeds and what they mean. We will get information from the accelerator trials, but obviously just given their age, it'll be a few years coming.
All right, John, do you want to keep on talking? How do we estimate max SDI on a given site?
So I guess there's a number of options. I guess one is to actually put in trials on a range of your different site types, and probably to keep a higher stock treatment. So maybe think about 1,000 stems per hectare plant and leave stand. Just reflecting on the earlier silviculture breeds trials, and the fact that either Mike resisted the push to go to a lower stocking, or I think in some of those trials the late thinning that was supposed to happen in some of those 1,000 stems per hectare plots never happened. Actually gave us a really good assay for getting carrying capacity. There are some other sort of ways that you can try and estimate it, but I think from a practical forestry perspective, just leaving a corner of a stand here and there and just not thinning is probably a way to get some idea of what's going on. We want to go back and do more intensive data mining of the extensive data set that we have got. We have the advantage that we've grown a few rotations of trees. We've grown a tree over quite a long time. We've got to the end of the rotation, and to use those data to identify the sort of genetic and site factors that contribute to carrying capacity and make that information available to people.
Okay, we have a question around genotype. Genotype is a key driver of productivity. Every year I plant 100 to 150 hectares. Every year I ask for the best available genetics, but over 15 years now I'm still offered the same GF19 and GF23 seedlings. How can improved genetics from GCFF be accessed by smaller forest owners?
So, what we've considered when we were approaching the stock that we put in, we tried to cast our net pretty far and wide initially around the genetics we could put in, but we were really quite limited in terms of what we could actually get. However, the approach that we've taken is that we're considering these based on their traits, because from year in, year out, we haven't been able to source the exact same genetic material across the accelerator trials. However, when there has been a clone we can't get in that year, we get a different clone that has those same basic traits, so that we're comparing those. And again, the knowledge of clones is one thing, but if you consider the traits that we're looking at, we can freely make that information available. I think it is in various publications around what we're doing in the accelerator trials. You can talk to your suppliers and say, I want trees that have these kind of traits, because that's what we hope that's what we hope can come out of this. Now, I'm not sure how well that will translate to what a given forest grower might be able to get from their given nursery. I mean, GF19 is common in a lot of places, and a lot of people aren't necessarily going to take the plunge to invest in developing up and producing another type of material. But again, that's one of the legacies we're trying to create with the accelerator trials, is to show that what you do, the selection from the nursery, and also even just the management in the nursery as well, they do have impacts on the life of the rotation through time, as well as how those trees will respond to management in the forest as well. So essentially, there isn't an easy answer to that question, but we have tried to structure it so that it's not about this clone, it's about a tree with these traits.
I think also too, that's probably a good question to bring up when they have the genetics session tomorrow, but also, hopefully with some nursery growers in the room. But it's probably a question also around forward planning and just collaborating to get scale, which probably can't answer now, but I know it's one that comes up. I hear that comment quite often. So I think it's probably worth raising it again tomorrow.
All right, did we have another? What does the financial return on Bio-ret look like?
It's an evolving proposition, and I probably shouldn't say too much more than that, other than the fact that it hasn't stopped us so far.
Okay, I think we're caught up there. So if we have any more questions from the floor for these guys? If not, I think we have about 4 minutes, so we'll give you a similar question to what the last group ended with. What do we have to look forward to the next 5 years with the accelerator trials?
Sorry, it's sweaty.
Well, I guess in the microbial space, we're going to continue building on the information that we've got. We're going to really expand the types of genes that we're looking for, so that we can continue to stack that productivity. So there's lots of trials. The accelerator trial, there's still some long-term trials that we can use the data from to help build the picture for us and really moving forward.
I think from my perspective, I'm really interested to learn more about stand dynamics for the range of genotypes that are on site. And obviously, in the next 5 years, we're probably going to reach some of our early thinning decisions. So really thinking about what we do there in terms of taking advantage of the opportunities and actually putting into practice everything we've talked about from a theoretical perspective.
I guess I'd just reflect on the Rangipo workshop that we had the other month, where we actually went to that stand. It was 3 years old, and you could walk through the stand and you could visibly see the difference in the clones, and also the difference between the stocking and ripping and non-ripping in terms of growth. So I think just getting the industry out there and having a look and getting them engaged, and that also links to helping us plan for what treatments we're going to apply, because the accelerator trials are going to the end of the rotation, and they're designed so that we can continuously have interventions on them. So it's not just one intervention and leave. It's like having a little heart monitor on. Okay, we've done one intervention and we've stacked up the productivity a little bit. So now let's keep monitoring that site and keep just putting on the little interventions to actually reach that site's full maximum capacity.
I'll grab about the only thing that's left. I'm really enthusiastic about working with them over the next 5 years to see what we can learn to build resilience, to build the tolerance across the national estate? What lessons can we learn from these 6 sites around how we can make them more resistant to disease, deal with adverse weather conditions, or alternatively make the best of better weather conditions? How we can take what we learn from these accelerated trials and make our national estate more flexible, more adaptable, so that everyone can benefit from what we learn.
Fantastic. All right. Well, I think we've made it to afternoon tea. So thank you again for contributing questions. And let's thank our speakers again for fielding them so well.